All right, so today we switch gears from talking about pedigree analysis, transmission of traits from one generation to the next, to finally the last part of molecular genetics. How do the chromosomes that we inherit get turned into proteins that affect our traits? So that's what we're going to start doing today. We continue for the next week or so. It's all Dogma, transcription and translation. So these are the important concepts for both this chapter and the next chapter, the chapter on transcription and the chapter on translation. We've seen how DNA passes from generation to generation. Now, what happens to the DNA when it's in the cells, the nucleus of an individual? How does that affect them? Especially, how do mutations affect the individual? These are important concepts for you to understand to get out of the material we're going to talk about this class and for the next week or two. You'll find on Blackboard that there's a PDF that looks like this, just a screenshot. You don't need to be able to see the details yet. You can download the PDF of this exercise. This exercise we're going to slowly fill in as we go through the next couple of chapters. So the point is, this is an image of both strands of DNA of a gene and the corresponding messenger RNA, the transcript. And the goal is by the end of the chapter on transcription and translation, you're to be able to answer the 8 questions that are there at the bottom. So download it to your tablet at some point. You don't need to do it right now. But by the end of class, we will be able to answer some of those questions already. So we're going to talk about the central dogma. DNA turns into, it doesn't turn into, but codes for RNA, which encodes for protein. The first thing we need to know is, how does the cell know where its genes are? Here's a drawing of a chromosome, like many of the drawings you drew for us already in class. How do these authors know where to end that drawing for gene 1 and start the drawing for gene 2? Here's gene 1, here's gene 2. They're drawn, and they are discrete objects, discrete parts of the chromosome. We can say where a gene starts, where a gene ends, where the next gene starts. How does the cell know? How do we know? What defines the start and the end of a gene? That's what we're here to find out. So the structure of a gene, right, starts with a promoter. Promoter is part of a gene. The promoter does not turn into protein, though. So this overview down here in the bottom goes through the whole central dogma. We start with DNA. It encodes a messenger RNA, a transcript, and that encodes, after translation, the protein, the actual functional unit, something that goes and does something in your cell, confers phenotypes. So the promoter is not part of the transcript, right? The promoter is missing in the messenger RNA. But that's where the gene starts. The promoter sequence. So it's not something that gets turned into protein. It's a functional part of the gene. So why is transcription and translation so important? What does transcription and translation do for us? Right, so if we have no control, thank you very much, it controls what genes are turned on. Every one of our cells, like in these slime molds that my father took a lovely picture of, right? Slime molds are some of the earliest multicellular organisms to evolve. They're groups of cells that have different roles. So up here, some of these cells have turned into a stalk. These look kind of like bean sprouts or something like that. There's a long stalk, and then there's this red cap on top. And the red cap is composed of cells that are gametes. They're essentially cells that are going to go out and make the next generation of this mold. And all of these cells, just like in us, have the same DNA in every cell. Right? My toe cell, my eye cell, my lung cell all have the same genes. How is it that those cells are different types of cells? So this is the concept of genomic equivalence. That every cell in our body has the exact same genes in it. So how how does my eye cell know to produce that crappy red opsin gene that doesn't let me detect red light, yet that red opsin gene doesn't get turned on in my toe? Obviously, my toe doesn't need to be able to detect red light anyway, but that's the point of transcription, is that every cell in our body turns on specific specific sets of genes out of the 30,000 or so genes that we have in all of our chromosomes. So the study of transcription is really trying to figure out how does a cell know and how does it control which of its genes get transcribed. Because I guarantee you there is no cell in the body where all 30,000 of our genes are all being transcribed. Different cell types turn on different suites of genes. And after transcription, this molecule, RNA, is produced. You may be familiar with the concepts. I hope you are, because I'm going to breeze right past this. RNA has uracil, U, instead of thymine, T. Okay. And the RNAs that are produced are single-stranded molecules. So they don't have, it's not double-stranded like DNA is. And what that means is that RNA molecules occasionally tend to base pair with themselves where they can. So like DNA, RNA is more stable when the hydrogen bonds between nucleotides can be satisfied, when it can base pair with itself. So RNAs aren't just linear single-stranded strings. They often fold up into complex shapes in the cell. And that's important for the function of some RNA molecules. The textbook talks about lots of different categories of RNA. These are categories that we geneticists have come up with. The two we're going to focus on, you don't have to know the rest. The two we're going to focus on in class this chapter and next are the transfer RNAs, tRNAs, three, ribosomal RNAs. tRNAs and rRNAs are important in translation. So we're going to look at them more next chapter. And the main category, which you've probably heard about a lot, messenger RNAs, which we also call primary transcripts. And what we're going to start doing today is figuring out how prokaryotic transcription and eukaryotic transcription differ. So how do bacteria transcribe genes? How do we transcribe our genes? What are the differences between them? What are the similarities? One of the differences is that we do things to our primary transcripts that prokaryotes don't do. We're more complex, not surprisingly. So we're focused on RNAs for this chapter. We're focused on this process. Transcription. The act of taking something written and writing it again, reading the DNA language and writing it into the RNA language, like this lady probably was not doing back in the day. But this process uses, like DNA replication, uses DNA polymerase. When you produce a messenger RNA, you use RNA polymerase. Same concept, it's an enzyme that links together ribonucleic acids, nucleotides that make RNA. So now we go into more detail. This is going to look exactly the same. You're going to see this slide again next week, as boring as hell as it is. Because translation is exactly the same way. If you're going to transcribe a gene, if you're going to translate a gene, you have to know where to start, where to stop, initiation and termination, and then the process of the actual production of the messenger RNA or the protein. So we're going to go through those three stages in a little bit more detail. Questions at this point? One of the key things that you need to know about, yes, it does work, by the way, those of you that are posting on today's meet. It worked, congratulations. Right, critical concept. Some of the proteins that we have in our cells bind specific DNA sequences. Specific DNA sequences, not any DNA sequence, but those proteins go around our nucleus on our chromosomes looking for specific DNA sequences to bind to. That's important in transcription. That's how the cell knows where to start transcription. So I told you that there was this region called the promoter region. This is it in all of its enlarged glory. This is the region of the gene where the gene starts. This is how a cell knows where a gene is located. And what these scientists did was they took the genes. So here's a transcript in red. For some reason, it's wiggly. So the transcript, the messenger RNA that was produced from this gene, started at the codon, or at the nucleotide called +1. Critical nomenclature for transcription. Wherever the messenger RNA starts, wherever the transcript starts, that's +1. We'll see that again on another slide. Everything here in yellow, what's the point about all these highlighted nucleotides in yellow? These are all upstream, or to the 5' direction from where these genes all were transcribed, the +1 nucleotide. Why are these highlighted in yellow? What does it mean to be a consensus sequence? So you have 7 different genes. If you look down here where they're actually being transcribed, TXN transcription, are any of those 7 genes the same in terms of DNA sequence or RNA sequence? Do they all have different sequences? Column, if you look down each column of nucleotide sequence over here, are there any where all 7 genes have the exact same nucleotide in them? There are a few that are close, 5, 6 out of 7, but there are none that are identical. But when you look in these yellow boxes, what's the pattern? All of these 7 different genes, regardless of where they came from and what their job is in the cell, seem to have the same sort of sequence, much less so over here on the left side, but definitely here on the right. Most of the genes have something that looks almost like TA-TA-AT. And that's how geneticists first realized, hey, just before where transcription always starts in bacteria, there's pretty frequently, almost all the time, this specific group of 6 letters, DNA nucleotides in a row, TA-TA-AT. So that's the signal that bacteria use to say, hey, there's a gene nearby. So there are these 2 consensus sequences, which basically means average consensus. We look at a bunch of different people's opinions, we develop a consensus, that's the overwhelming majority. So these consensus sequences down here are generally the DNA sequences that are upstream of genes in bacteria. Why is it important that there are specific DNA sequences? What does that do for transcription?
Makes it easier.
How does it make it easier? So there's a specific DNA sequence. Big what? Pardon? So it's something. So what's recognizing this DNA sequence? It could be RNA polymerase, the thing that's actually going to transcribe the gene. Who needs to know where the genes are? The thing that's going to read the genes and turn them into messenger RNA. So here's a little bit of terminology you've already read, so I'm going to go over this pretty quickly. Each of these single strands of DNA from the different genes is written from the 5' end to the 3' end. That's just the convention, like we always do. We write genes 5' to 3', left to right. And so the promoter, as it's drawn here, is always at the 5' upstream, which is another way of saying this, or on the 5' end of the gene. So we always write from left to right the promoter sequence, and then there's the nucleotide where transcription starts, and the gene goes towards the right side of the screen. And I've already mentioned a little bit about the numbering system. So the very first nucleotide that's part of the messenger RNA, the RNA molecule, is called +1. And +1, what comes to the right of +1? +2. Not a trick question. I should have told you in advance, not a trick question. Here's the trick question. What's to the left of +1? -1. So there's no 0. Sorry, so that's the trick question. So it's +1, +2, +3, +4, +5 to the right. And if you count backwards from -1, for some reason there's no 0 nucleotide, because that would indicate there's no nucleotide there, I guess. So +1, the one to the left of +1 is -1. And then you count backwards, -1, -2, -3, and so forth. So -1, -2, -3, that direction. So the promoter lies in the negative space. The minus nucleotides is where the promoter exists. And that's our just convention. That's how we discuss this. It doesn't have anything to do with the biology of the system. So there's two consensus sequences that we were just talking about. Right? You line up the genes and the promoters of these different genes in bacteria, and there are those two consensus sequences that are about -10 or 10 nucleotides upstream of where transcription starts, and about 35 nucleotides upstream or to the left side of where transcription starts. So that's the promoter region. Contains the -35 and -10 consensus sequences. So it turns out it's not actually RNA polymerase itself that binds to these motifs or consensus sequences. It's a protein that is associated with RNA polymerase that binds these sequences. And the way this works is there's a protein which is called sigma factor. Remember that, sigma factor. Sigma factor is the protein that binds the -10, the -35 sequences. So this is, like I mentioned earlier in class, it's one of these proteins that binds a specific DNA sequence. The protein itself, cruising around the nucleus, and every time it sees PTG, ACAT, and then there's a space of about 15 to 17 nucleotides, and then it reads TATAAT, that protein binds tightly to the DNA when it sees that sequence. Those two sequences about that distance apart on the chromosome.
And that starts at the promoter region?
And so this is in the promoter region. So sigma factor binds the promoter. Sigma is a protein that recognizes this promoter sequence, what's shown in yellow. Yeah?
Question, this is a bit unrelated, but so when we're like inserting a gene into a different organism, we're gonna have a different sequence for the -10, -35, and usually we'll put like a promoter or something, but then we need different sigma factors also, right?
So there are two questions contained therein. One is a question about sigma factors. So it turns out in bacteria, the way that bacteria control which genes are turned on and not turned on, because bacteria have lots of genes too, they have different sigma factors that recognize different sequences and turn on different genes at different times. So the sigma factor we're talking about is a specific sigma factor that recognizes this sequence. In bacteria, there are other sigma factors that recognize different sequences that are upstream in the promoters of different genes.
So they would be able to recognize sequences from different species, etc.
And then if you want, so the first part of the question was about making transgenic organisms. So a lot of times geneticists take genes from one species, put them into another species. If you take a gene from jellyfish and you inject it into a bacterium, what's not going to happen? I think we're talking about that. not going to be transcribed, right? Because the jellyfish gene probably does not have this DNA sequence that the bacterial proteins are looking for in the promoter of that gene. So if you want to take a jellyfish gene and put it into bacteria and have the bacteria actually produce the protein that that gene encodes, you have to, as you said, provide it with bacterial promoter. So you take a bacterial promoter, you put a jellyfish gene next to it, sigma factor binds to the bacterial promoter, and then what happens next after sigma factor binds the promoter? So somehow we have to involve RNA polymerase, the gene that's actually going to start transcribing the DNA. And basically sigma factor is like the GPS for RNA polymerase. So RNA polymerase is the car. You take sigma factor, the GPS unit. Sigma factor tells RNA polymerase where to go. So anywhere sigma factor is bound to a chromosome, sigma factor physically binds to RNA polymerase. There's chromosome stretching across here. I'm sigma factor. I bind to part of the chromosome, and then I grab RNA polymerase and say, hey, get over here. And so sigma factor recognizes the DNA sequence and binds to it. Another part of the sigma factor protein physically binds to RNA polymerase and says, hey, there's a gene here, it needs to be transcribed. So that's what sigma factor does. Sigma factor binds first, and then what we call, and I'll use this term again, so I want to tell you what it is now, recruits RNA polymerase. Sigma factor binds to the DNA and it recruits, it brings in RNA polymerase and says, hey, this is a gene, transcribe it. Just a different cartoon of the exact same thing. So to summarize, sigma factor binds here. In this case, speaking of different sigma factors that bacteria have, the specific one that's being discussed here is sigma factor 70, which probably means there's a 69 and a 68 and a 67 and so forth. So bacteria have lots of sigma factors. It binds the DNA first in the promoter, minus 35 and minus 10 sequences, and then it brings in all of the proteins that constitute RNA polymerase. Then sigma factor leaves. It stops binding the DNA. It's brought RNA polymerase to the gene, and RNA polymerase then starts transcription. So how does this differ from eukaryotic? This is all bacteria up to this point. Now let's talk about us for a second. How do we start transcription? Same but different. Instead of sigma factors, we have a protein called TATA binding protein, TBP. Right there. It's like sigma factor. It recognizes a specific DNA sequence that's in the promoter of a gene, it binds to it, and it brings RNA polymerase to the gene. There are a lot more proteins involved in eukaryotes than prokaryotes, but the concept is the same thing. One protein recognizes one sequence in the promoter, and then it drags RNA polymerase in and says, hey, start transcribing. So that's all initiation. That's all you need to know about initiation. How does a cell recognize where its genes are? What's the signal? What's the code? Shout it out. I'm ready. Not the specific DNA sequence, I don't care about. What defines the start of a gene? What's to the left of the +1 nucleotide? The promoter. Right. And the promoter contains these specific DNA sequences that either, if you're a prokaryote, sigma factor binds to, or if you're us, tau binding protein binds to. So at this point, RNA polymerase is on the DNA, ready to start making RNA. That's the process of elongation, the second step in transcription. So RNA polymerase has many functions, one of which is it melts the DNA, just like replication. It separates the two DNA polymerase and replication. Here, RNA polymerase in transcription melts the DNA to create a transcription bubble, and then it starts using base pairing to match up DNA nucleotides with the appropriate RNA nucleotides. So RNA polymerase synthesizes, sticks together the nucleotides that produce the RNA molecule. So like DNA synthesis also, RNA synthesis in both prokaryotes and eukaryotes is exactly the same. Starts at the 5' end, RNA polymerase synthesizes from 5' to 3', just like DNA polymerase does. Which means at the end of the RNA that sticks out first from RNA polymerase is the 5' end. The 5' end of the molecule of RNA is the first nucleotide that gets produced. So as RNA polymerase slides left to right, the RNA molecule that is producing exits RNA polymerase, and the 5' end of that RNA molecule is hanging out there in the nucleus. So a little bit more terminology. Here in the drawing on the bottom, what's another name for the non-template strand of DNA? Pardon? Coding strand. So there's important things here to realize. RNA polymerase only reads one of the two strands, as was shown in the previous cartoon on the previous slide. It only reads one of the two strands. That's where it uses base pair complementation to match an A in the DNA with a, what nucleotide would that be in the RNA? If there's an A on the template strand, it's going to be matched up by RNA polymerase with a U. Okay. So RNA polymerase is using the template strand to pair up with the appropriate RNA nucleotides. So if the template strand, which is the strand that RNA polymerase reads, is complementary to the messenger RNA molecule, and because these are two strands of DNA, the template and the non-template strands, or the template and the coding strands, they're obviously complementary to each other because they were the two strands of DNA. So what does that mean about the RNA molecule and the coding strand? Their sequences. They're the same. If the template strand base pairs with the mRNA and the template strand base pairs with the coding strand, then by definition, the coding strand and the messenger RNA sequences are going to be identical. Except, what was that?
The diamine in place of yours.
Right, so except for that there will be U's in the RNA and T's in the DNA. Otherwise, the coding strand and the messenger RNA will be identical in sequence. So an important thing to remember. So that's how you distinguish coding strand from template strand. Template strand is always going to be complementary to the messenger RNA, which means the coding strand and the messenger RNA are going to be identical in sequence. With that in mind, take a couple minutes, discuss with a neighbor if you like. If you've got questions, either raise your hand or feel free to post them on today's meet while you're trying to decide the answers to these 4 questions. Okay, we'll reconvene in about 3 minutes after you've had a little chance to work on this. Pardon?
Would the template strand be the one that the coding strand is copying?
So here's the messenger RNA molecule. So which strand is it going to be base paired with? So is this the template, or is the blue arrow pointing to the template strand or the coding strand? The messenger RNA is going to be complementary to what it's base paired to. So is it template or is it coding? Look back at the previous slide if you want. The messenger RNA is complementary in sequence to the template strand because it's the template strand that's being base paired with the RNA nucleotides. And that means the opposite strand is the coding strand. The messenger RNA and the coding strand will both be identical in sequence except for U's and T's. If you looked back farther, which end of the RNA is hanging out there into space? It's the 5' end, because it's the end that gets synthesized first. So polymerase is actively working on synthesizing, adding new nucleotides at the 3' end of the RNA molecule. Does this help us figure out the polarity of the DNA strands? What's the polarity of the template strand? What's here on the left side? That's going to be, let's see, here's a double-stranded hybrid DNA RNA molecule. So it's going to be anti-parallel like all double-stranded sequences. So if the 5' end of the mRNA is on the left, what's on the left side of the DNA strand it's hybridized to? 3'. So this end must be the 5' end of the template strand, and then of course the coding strand is anti-parallel to that. Finally, which direction do you think polymerase is moving? Going left to right or right to left? So that 5' most nucleotide corresponds with nucleotides down here. That was the first nucleotide to be incorporated right here. Now polymerase currently is right there. So it's moving, started on the left side and it's going to the right. Polymerase moves in that direction. What polarity does that? Don't remember this. I'm just going to point this out for those of you that remember this from DNA synthesis. What direction does that mean that the polymerase is moving on the template strand? It moves on the template strand from 3' to 5', while it synthesizes 5' to 3'. Exactly like DNA replication. Don't need to memorize that, but in case that helps you remember, it's just like DNA replication. So that's most of elongation. RNA polymerase moves along the template strand, adding in complementary nucleotides. That's true in prokaryotes and eukaryotes, but we do something fancy, which means you've got more work. Understanding the process of, can I say that? The process of processing transcripts. So this thing called processing is only something that happens in eukaryotes, in us, not in bacteria. And it's got 3 steps: capping, splicing, and tailing. So there are going to be 3 different types of modifications. Our cells take the first molecule of RNA that gets produced by transcription, modify it in 3 different ways. Something happens to the start of the transcript, the cap, something happens to its tail, and something happens in between. And this is just a comparison of how this works in bacteria on the left, where there is no nucleus, versus us on the right, the eukaryotes, where we've got a nuclear membrane. Presumably, the reason that we do transcription is because somehow information has to get out of the nucleus, where our genes live, into the cytoplasm, where translation happens in eukaryotes. Right? There's that nuclear membrane that stops the ribosomes, the things that produce the proteins, from interacting directly with our genes. So the transcript, the RNA, is important because it sends the message from the nucleus to the cytoplasm. It brings the information to the cytoplasm where translation happens, and that's what we're going to look at next chapter. So initially, key point here, initially, upon transcription, we've got the transcript. We call the primary transcript before all of these events happen. After splicing, capping, and polyadenylation, tailing, we refer to that finally as the messenger RNA. So there's a difference between primary transcript and mRNA, or messenger RNA. And that difference is, has it been processed yet or not? So a different way to look at the same thing. We've got a stretch of DNA that contains the ovalbumin gene. Here's the primary transcript. What's different between, just qualitatively, in your terms, not being exact, what's different between the DNA and the primary transcript in this case, if you just compare the 2 lines? What's really different? It's shorter, not by very much. Okay. There's a little bit of the part of the gene, so that's what's drawn here. That's the gene. Primary transcript's a tiny little bit shorter. But then what's the difference between the primary transcript and the processed or mature messenger RNA? What's happened? We've lost all of the colors that represent introns. So all of the introns get removed. That's splicing. Although it's not necessarily all of the introns that get removed. That's a complication we have to deal with. What else is different between the primary transcript and the mature messenger RNA at the bottom? Aside from we've lost the intron colors.
The cap and the tail.
Yeah, a cap and a tail. So physical modifications of the messenger RNA at its ends. Something's been added to the 5' end, the cap, and a tail has been added to the 3' end. Graphic representation of the 3 steps of processing. Please do not feel obliged to remember the details of capping. The only thing I want you to remember about capping necessarily is that what the cap is. What is this physical structure that's attached to the 5' end of a transcript? It's a nucleotide. It's a special type of nucleotide, but that's all it is. It's a single nucleotide that gets stuck on the 5' end of the transcript. The nucleotide happens to be 7-methylguanosine. That's it. Sometimes abbreviated 7-MG, 7-methylguanosine. What this cap does is what's really important, which is that second bullet point in the middle here on the bottom left. Protects the messenger RNA from being chewed up by enzymes in the cell. Yes, sir?
Well, wouldn't it also have to have, like, because it has to pass through the membrane of the nucleus, wouldn't it also have to have, like, some kind of way to pass through it? Wouldn't the cap also help with that?
Yes, so the cap does also help with identifying this transcript, this RNA molecule, as something that should be sent out of the nucleus into the cytoplasm. So the machinery that's in the nuclear membrane that recognizes transcripts and pulls them into the cytoplasm also grabs onto that cap. That's how it knows, this is an RNA molecule that's been processed. It can now be safely moved into the cytoplasm. Splicing is the most complicated of the three steps. Go ahead. Yeah.
Can you go over the overall steps of this process real fast?
Which process?
This process.
Splicing?
Yeah.
So, yes. I don't expect you to do the biochemistry. That's for biochemistry class or organic chemistry. I'm probably going to use words like nucleophilic attack or something like that, but I'm going to hate myself for it. We're going to go over splicing much more in detail next class. So here's the overview, though. And I was, after all, a chemistry major, so that's why I use words like nucleophilic attack. Splicing is simply the removal of introns. We're going to talk more later about what an intron is, how it gets recognized. But all splicing is taking two pieces of messenger RNA and sticking their ends together while cutting out something that's in the middle. So here was originally the initial transcript, a pre-messenger RNA, that has a piece of RNA that's going to be taken out. That's how we define it as an intron. Anything that gets removed during splicing is technically an intron. Okay. Here's how the introns were initially discovered. We lined up, and this is a screenshot from the PDF exercise that you should download from Blackboard. You line up a messenger RNA sequence here with the DNA sequence it came from here. And what scientists very early on, decades ago, realized was that the messenger RNA molecule, if you line it up with the DNA, has these gaps in it. That is, the gene that encoded this messenger RNA molecule is longer than the messenger RNA molecule itself. There's something missing from the transcript. It's there in the DNA, but after transcription, that sequence of nucleotides is gone. That defines this region as the intron. So then the question has to be, well, how does the cell, like transcription, how does the cell know which parts of the gene? Holy crap. Genes are important. They encode our proteins. And now I'm telling you that there's proteins in our cells that take transcripts and break them into bits and stick them back together. You better believe that's a process that's very tightly regulated, because if you screw up splicing, you screw up the messenger RNA that you produce, you screw up the protein you produce, you get some sort of disease disorder phenotype. So here's how the cell knows what is an intron. It's exactly the same as, well, it's similar to the promoter. There are consensus sequences. At the 5' end of every intron, this is the part that gets taken out. This whole thing right here is an intron. The 5' end always starts with a G and a U. So those numbers down there below the G and the U are the percent of time those nucleotides are found at the start of an intron. So 100% of the time there's G, 100% of the time the next nucleotide in the intron is a U. At the other end of the intron, the 3' end, the last two nucleotides that get taken out, guarantee you, sure bet, A and G. And then there's also this weird A, the branch point nucleotide, in the middle. So there's going to be an A somewhere in between. So that's how you recognize an intron. GU starts the intron, AG ends the intron. Now in our last 45 seconds together, and this is the chemistry. You don't have to know this. But the reason those GUs and the AGs are necessary is because those nucleotides wind up getting bound by proteins that stick the GU and the AG together into the piece of the messenger RNA, the intron that gets released after splicing. So next class we talk almost exclusively about splicing. Here's why splicing is so doggone important. Our genes have more than one intron, which means that there are more than one way to splice, there's more than one way to skin a cat, there's more than one way to splice a gene. So in this example, there are what? 5 exons. So in between, the exons are the parts that remain after splicing. So that means there's an intron, intron 1, intron 2, anything between exons is an intron. So here there are 4 introns in this gene. Sometimes introns aren't removed. Sometimes introns don't get spliced out. Sometimes by accident, an exon might accidentally get removed along with a couple introns. This is the concept of alternative splicing. One gene does not always get spliced the same way, one transcript, one messenger RNA molecule doesn't always get spliced the same way every time. So we're going to pick up next class with alternative splicing, and what I'd like you to do is to just do some simple mathematics. Here are 3 exons, so there are 2 introns. There's exon 1, exon 2, exon 3. There are 2 introns here in red. If you were how many different ways can you splice this gene? So see if you can come up with the maximum number of ways you could splice this gene. And we'll see each other on Monday. Have a fabulous Halloween.